Hello, Eva Professor and the research. First, I would like to thank Pencis to provide me with this chance to introduce my recent research. I'm from Chengdu University of Traditional Chinese Medicine in China. This is a comprehensive university that focuses on all aspects of traditional Chinese medicine related fields. In our team, we have recently designed, synthesized, and evaluated a nucleic acid drug conjugate. Because of its specific targeting modality and superior therapeutic efficacy, and therefore we boldly predicted that this kind of conjugate may be the next generation of targeting and tumor drugs. My presentation contains 10 parts. First, let's take a look at the current changes of pancreatitis. We know pancreatitis is one of the most frequently used first-line drugs for cancers. However, it has two severe drawbacks. One is its toxicity. Pancreatitis has no tumor targeting property, and it can't distinguish between normal cells and tumor cells, so it will lead to undesirable side effects. And another drawback is its poor water solubility. It is almost insoluble in water. Currently, the product Taxol utilizes chlorophyll EL as a co-solvent. However, the co-solvent could usually cause hypersensitivity. Another pancreatitis-like drug launched in 2005, named Orbexon. Indeed, it improved water solubility, but Orbexon as a complex is difficult to process and control quality. Moreover, some studies recently reported that it could increase toxicity of pancreatitis. Now, facing with these questions, we want to develop a pancreatitis derivative. It will not only improve the water solubility of pancreatitis, but also make it have tumor targeting property to reduce its toxic side effects. So we got an innovative strategy that we want to design an optimal drug conjugate. Due to the special property of the optimer, we hope the conjugate has good water solubility and tumor targeting property. So, we know optimers have special three-dimensional structure formation. They can specifically bind to their targeting molecules. They also have other properties, such as good water solubility, quickly and reproducible synthesis, and high affinity, excellent stability. So, we think optimers are the suitable choice to compensate for the drawbacks of pancreatitis. We know in normal cells, the nucleolin and the kind of protein mainly locks in the nucleus, but it overexpresses on the cell surface in cancer cells. The optimer S1411 could target nucleolin protein, and its specificity and safety were proved in clinical phase 2. Now we need to link the optimer to pancreatitis. From the viewpoint of chemical synthesis, The second hydrox group is most reactive function group, but from the result structure-activity relationship, we know that the second hydrox group in pancreatic cancer is an important bioactive site. And if the group is permanently modified, the obtained pancreatic cancer derivative will have reduced or lost the biological activity of the parent pancreatic cell. So the requirement remains as a label dipeptide linker. The dipeptide linker consisting of vanillin and citrulline can be broken by quercetin B. In the environment of blood circulation, quercetin B can't exert its biological activity, so this dipeptide linker can exist stable in the blood circulation, while in the microenvironment of tumor cells, quercetin B, which is present in large quantity, features biokinetic activity and can specifically broken dipeptide linker and release the prodrug pancreatic cell after a safe elimination reaction. After the structure design, we commence to synthesize the conjugate. This is synthesis route. Step 1, we synthesize the dipeptide linker. Step 2, we link the dipeptide to pancreatic cell and form the pancreatic cell derivative. Step 3, we conjugate the pancreatic cell derivative to the optimer and obtain the nuclear optimer pancreatic cell conjugate. Use this same method, we synthesize other conjugates, including fluorescence labeled conjugates and double fluorescence labeled conjugates. After that, we analyzed and characterized the purity and quantity of the obtained conjugates by HPLC and mass, respectively. All the obtained conjugates are our target products. Subsequently, we further confirmed the conjugate structure by one-dimensional NMR and two-dimensional NMR. After then, we tested the water solubility of the conjugate from the experimental results compared to the data provided in or reported literature. We found that the conjugate has improved the water solubility of pancreatic cell more than 1,000 times. To further verify the sensitivity and the stability of the dipeptide linker, we introduced the Fred assay. According to Fred theory, when the linker is intact, the fluorescence of both fluorophore in the conjugate remains stable. When the linker is broken in the presence of quercetin B, Fred is interrupted and the fluorescence intensity of the dam is decreased while the film is increased. These are fluorescence emission spectra of 3 different compounds. In the presence of quercetin B, the intensity of double fluorescence labeled conjugate is changed over time. In human serum and PBS, the relative fluorescence intensity and fluorescence intensity remain almost constant. In PBS contain quercetin B, the relative fluorescence intensity as well as the fluorescence intensity and 519 nanometer granularly diminished with the extension of time. It could suggest that the dipeptide linker in the candidate was catalyzed by quercetin B. To examine whether the modification of pancreatase affects the affinity between aptamer and the nuclear protein, we employed the molecular dynamic simulation and the ITC assays. The experimental results displayed the affinity between aptamer and the nuclear protein was basically unaffected before and after pancreatase modification. In the three different cells, we found that cells with high amounts of nuclear protein expressed outside the cell membrane took in more NP conjugate. When cells were co-incubated with aptamer RNA, the uptake of NP conjugate by cancer cells was significantly reduced. Then we tested the effect of modification of similar uptake of conjugated pancreatase. We found in SKOV3 cells, the aptamer the nuclear aptamer modification could facilitate the uptake of conjugate pancreatase in a time and concentration dependent manner. And the nucleic acid drug conjugate, we found that the conjugate entered the tumor cells by the micropinocytosis. The further trace of this conjugate revealed that it eventually reached to the nephron or quercetin B rich organelle to facilitate the blockage of dipeptide linker to release pancreatase. In terms of antitumor activity and toxicity, NP conjugate showed almost the same antitumor activity as pancreatase against two different types of ovarian cancer cells. However, the toxicity of NP conjugate was significantly lower compared to pancreatase. With regard to the effect on cell cycle, both NP and pancreatase cause cells to arrest in G2M phase, indicated that it was still pancreatase that played an inhibitory role in NP conjugate. 2 hours after injection conjugate, the fluorescence intensity from NPR in tumor tissue was quite higher than from CPR, and 4 hours later, the intensity from NPR in liver and kidney was remarkable lower than from the CPR. The section also showed tissue distribution of conjugate. All the results indicated that the NPR has adverse targeting properties to tumor, while NP conjugate is mainly metabolized by liver and kidney. After 3 weeks administration, NP conjugate showed a remarkable tumor suppression compared to the other group. The inhibitor effect of NP conjugate on tumors was further demonstrated by stain antigen Ki67 associated with tumor cell proliferation as well as tumor tissue section. After 3 weeks administration, the body weight of mass in NP group was similar to that of PBS group, while the body weight of mass in the pancreatitis group was almost significantly reduced. Meanwhile, after testing the leukocyte count, it was found that the changes in leukocyte count before and after treatment in the NP group was similar to that in the PBS group. All these above results can indicate that NP could significantly reduce the toxic effects of pancreatitis on mouse. Fairly HE study of the main organs of mice reveals that both the pancreatitis group and the CP group have the same toxic effects on the heart, liver, spleen, and lung, while the NP group only slightly affected the lung, so the modification of targeted optimal could help reduce the toxic effects of pancreatitis on normal organs. To make a summary for this project, we designed and synthesized the nuclear optimal pancreatitis conjugate. It has good auto stability and storage stability. The nuclear optimal modification improved pancreatitis and tumor activity and reduced toxicity. This work established an innovative technology platform for optimal drug conjugate development. Moreover, the personalized pancreatitis conjugate could be developed for the patient by altering optimers. Importantly, this work provided an efficient approach to the next generation of smart and tumor drug discovery towards precision medicine. Thank you for your listening.